And in the last part for today, we will have a talk given by Thomas Beyer and Laliat Kumar. Thomas Beyer is full professor at the Medical University of Vienna, where he leads a quantitative imaging and medical physics team. He is co-developer of the first combined PET-CT systems worldwide, and he has a background in research and project management in academia and imaging industry. And lately, Thomas engages in the exploration of the human functional connectome using total body PET imaging. Laliat is a postdoctoral researcher in the Thomas groups. During his PhD studies, he's established a clinically viable computational framework to determine the cerebral metabolic rate of glucose in the fully automatic and non-invasive manner by using the synergetics information provided by the integrated PET and MRR. He is currently developing data analytics schemes for probing the human metabolite connectome using extended field of view coming from the PET-CT scan systems. So I'm really looking forward to their talk.
I think Thomas is not able to share his screen because he's not a panelist yet.
Oh, thank you. We will look into that.
I think he should be a panelist now.
All right. Typical question in COVID times. Can you hear me?
Yes, we can.
Yes.
We cannot see you.
Thank you so much. I think after this kind introduction, we should rest the case and just conclude with the same conclusion as the previous speaker. We're hiring. And I think there's not much else to say. In fact, all the credit goes to Lalith, who kindly prepared this slide. And he asked me to introduce the simple stuff in the beginning. And also we had promised to Renata that I would have at least a verbal appearance. So here I am. Lalith is part of our joint team. We actually handle it differently. It's not named after the most senior guy, but it's actually given a name which we call quantitative imaging and medical physics. Now, having followed the talks today, I must say we feel a bit like outsiders. So we want to actually bring your attention from a single cell omics approach to four-dimensional molecular imaging and quantification. But moreover, we're kind of outsiders because I'm from a small town in East Germany and I'm a guy with wild hair. Thank you, Lalith, for making this icon. I will love you too. And then
Thomas, I'm sorry to interrupt you, but I don't think we see your slides.
Okay. This is most unfortunate, Lalith, my dear friend.
I don't think you are. Now we see it. Yes, perfectly. Thank you.
See, you should actually never invite old people to these type of meetings again. So I will stop repeating myself, but these are the slides that Lalith made in the beginning at least. And I'm from East Germany. Lalith and I met about 5 years ago. He joined our team as a PhD candidate at the time and has moved stellar in his career. We both like food. I don't know why Lalith only shows it for himself, but I think it's a nice match of being a medical imaging physicist and a biomedical engineer. The objective of our talk today is really to share with you our passion for PET imaging, which is a molecular imaging modality, and homeostasis as a basic principle to understand diseases better. We want to share with you, and this is really where Lalith is jumping in, our attempts to visualize homeostasis and perturbations thereof, which are indicative of diseases. And we fondly feel that molecular imaging by the use of PET will be playing a major role in this, in addition to all other functionalities that you've heard about today. And finally, we're particularly glad that we were invited to this meeting because we want to finish with a plea for you to help us as we got stuck in some of these aspirations. So PET, I'm not quite sure how much you know about this. Probably a lot of you have these fairy creatures at home, call them PET. We love them because they give us joy. They love us because we give them food, with or without chicken, doesn't matter. But this is really not the PET we're talking about. We're talking about the PET as it stands for positron emission tomography, and it creates these fuzzy looking images to the left, which you should not get shocked by because they contain a lot of information. And since about 20 years, they come intrinsically combined with CT, which you see to the right, and which I'm sure you're very familiar with. And they are so-called PET-CT images or dual modality or hybrid images. The reason why they're combined with the CT is very simple, because it provides underlying anatomical information to what we call specific molecular information. Now, even though we heard earlier today that most of you are PhD candidates, I'm sure you've seen enough medical images to help me with this case study. So this is a case study of a patient suffering from disease that you see on the CT of the thorax. And on the right-hand side, you see the PET image. Can you tell me, or can you think of what's wrong with that patient? I'll give you 10 seconds or 5. So my question is, what is wrong with this patient? And the answer is very simple. The patient is dead. And why is that you can make this diagnosis? Simply because what I show you as the PET reference frame is empty. There is no signal. And this is really the basic of PET as well as nuclear medicine imaging. Unlike in radiology, we don't use radiation that comes from outside and penetrates the body, and we measure attenuation, but we actually target the living organism through the help of biomarkers. And these biomarkers are biomolecules that are labeled with radioactive isotopes, radionuclides, as you can see in the top left. You see a sugar molecule with a radioactive antenna. We inject those into the patient, they distribute inside the patient so as to create the bespoke images you've seen in the previous slide. So the key message for you is really that with molecular imaging or nuclear medicine imaging, we target the metabolic and signaling pathways in the body. And this tumor would have been seen had the patient be alive through an increased accumulation of this labeled sugar molecule, because cancer cells are hypermetabolic and divide at great speed. So once these radio tracers, and this is just one example with the labeled sugar molecule, you can label amino acids, water, what have you, depending on which metabolic pathway you'd like to probe. Once they are injected into the patient, the radiation is emitted from the redistributed tracer molecule, and it's being detected typically by rings of scintillation detectors arranged around the patient. Normally, and depending on your budget, the axial field of view is an order of magnitude longer than in CT, but certainly not yet covering the entire body, as you will see in just a short while. And out of this collected data from the emission scan, we reconstruct the radio tracer distribution. And not only we see these beautifully colored images, in this case, it's FDG of a brain with an axial slice, but we can also quantify these concentrations of the tracer in the different tissues and organs. So through the use of PET and the tracer principle, we are able to quantify physiology. Now, each modality has its own history. Ours started in the 1950s with people doing a rather rough setup on a workbench here. The first commercial systems came out in the 1980s, and PET has then transited into the field of hybrid imaging in 2000 with the introduction of a merger with the CT, or 10 years later with the merger of an MRI, which is considered a technical revolution, but a medical evolution. But nonetheless, you can see the different colors and tastes or flavors that PET comes in and is now available. Just to give you a ballpark figure, worldwide, we have about 6,000 PET-CT systems and PET-MR systems, compared to about 10 times more for the CT. Now, what we're really excited to share with you is new concepts that relate to the latest innovation in the field of PET. And this is the so-called Explorer system put forward through a collaboration with American colleagues in UC Davis and United Imaging. And this system is a PET system that allows to cover the human body in total with 2-meter axial field of view. So you can have a full total body image as you see to the right of the system. And why this is interesting and why this is a breakthrough invention, Lalith will go about, except to say that we want to mark up on the extended field of view that is unique in the field of diagnostic imaging, comes with a high, for molecular imaging people, spatial resolution, and most interestingly with a unique high temporal resolution in the sub-second arena. And this will add to the armamentarium of using biomarkers along the time domain as well, as you will see in a short while. So all these three things combined will give us the tools and functionalities to visualize and quantify inter-organ communication in the context of homeostasis and diseases. And with that, the simple part ends, and I will ask Lalith to take over the really cool stuff.